It's been great. It's been a great series of speakers, but I mean, we're finishing strong this time. Very strong. All right, just a little bit of a reminder for the students in the class, sign the paper. I'll say it every single time, and every single time someone forgets about it. You also owe me a reflection, a final reflection, and the discussion on the, been a long day. On ELC discussion. Okay, yeah. I'm still there in the log. And so please, I'll send a reminder via email, but some of you already sent me things, and I'll be reading those that are very interesting. So keep sending me stuff. And if you don't, I will remind you a couple of times, and then, I'll fail the class. Sorry. There you go.
When you say sending anything, you're supposed to like.
It's on ELC.
Okay.
Like,
so just on
but then I receive it as, as an email. And some of you were like, I'm not sure if it got it to ELC, and also write me an email. It's the submission is on ELC, but then, I receive all of that. Yeah. Said that, I will have Dr. Sai introducing our host for today. Thank you so much for showing up.
Welcome, everyone. It's my great honor to introduce the speaker today, our very own Merkle. We probably said the best for the last, or said the most senior for the last. For the semester, Scott joined the faculty here in 87, right? Almost 4 decades of service. I think among the current faculty, Scott is the most. Yeah.
You could put it that way.
Scott has worked on many different things. I think today we're gonna hear some of the things that many of you may not know or remember. Most of you know Scott has been working on American chestnut and the restoration and such. I actually was in my office going through his long 20 pages long CV. I saw the very first chestnut grant and chestnut publication back into the early 90s, right? So it's really several decades of continuous work. Scott got multiple awards. Again, I don't have my notes in front of me, but one SAF Barrington Forest Memorial Awards for outstanding research in forest biology. I think he was presented an outstanding contributions, propagation, IU from IU for Working Party, and then a UGA Creative Research Medal several years ago, and just last week, Creative Research Awards for his research company. Actually, I know Scott way back. We were just talking probably the very first meeting I went to as a brand new assistant professor back in the late 90s before I moved here. So today we will hear some of the key highlights from Scott's distinguished career. So,
Wow, this is just like Denver, except everyone's older. Okay, well, thanks for coming today. I hope many of you were intrigued by the title. I tried to come up with the stupidest title I could, and you'll see, if I don't forget to point you to them, where Alice in Wonderland and Mel Tormé come into it. Some of you may already know. So before I get started, I want to talk about, and this goes way back, who I came here to work with as a postdoc before I was even on the faculty. These two guys were professors here in Warnell before it was called Warnell. Dr. Claude Brown was a tree physiologist, in fact, probably one of the top 2 tree physiologists in the country I know of at the time, and his book was very famous. And Dr. Harry Sommer was the first researcher to clone pine trees in tissue culture. I was very lucky to get a job as a postdoc with them, and I learned a lot from both of them. I want to thank Wade Newberry. I don't know if Wade's here or not. The only photo I could find of Harry was a black and white photo from the inside of a book that he had published with Claude. And it was a terrible photo. And I took it to Wade and I said, Wade, can you colorize this and make it look better? And it came out pretty good. In fact, Harry has more color there than he had in real life. Sadly, both of these great scientists passed several years ago, but they were great friends to me and great mentors. So a lot of what I'm going to talk about today involves a tool called in vitro propagation. And there are different types of in vitro propagation, and I'm going to focus mainly on one. But what it is anyone know what in vitro means literally? Except Aiden? Yeah.
In the glass.
It means in glass, So this is growing trees and parts of trees in tissue culture in test tubes, petri dishes, and these plastic boxes we call magenta boxes because the Magenta Corporation makes them. Micropropagation is something that's used a lot in horticulture to propagate a lot of plants, including woody plants. All it is, it's like making rooted cuttings except in sterile culture. You take shoots and you get them to multiply, make more shoots from their buds, and then you clip those off and root them in culture. So it's a very fast way to propagate stuff up that you want to propagate, mainly for ornamental purposes. In forestry, not so much. Organogenesis is kind of like that, except you can actually take a piece of a leaf and fool it into making buds right on top of the leaf where there aren't supposed to be any buds. And then you can do the same thing. You grow out the shoots and you root them to make plants. That's called organogenesis. And because the shoots are new, they're not from preformed buds. They're called adventive or adventitious buds. And the tool that we use the most is called somatic embryogenesis. And I will define that and show you what it looks like in just one more slide. But I wanted to let you all know this is all done in sterile conditions because we grow this stuff on tissue culture media, and I'll show you what the formula looks like for tissue culture media, which has sugar in it and lots of nutrients. And of course, fungi and bacteria would like to grow in there and just grow right over the top of our plant cells and plant organs. So it's all done, the work's all done in laminar flow benches or sterile benches. And we autoclave all our stuff and we wear gloves and we spray ethanol everywhere because if we get contamination, that's the end. We got to throw those cultures away. And so people get a lot of training to do this, and some people get really, really good at it. This is what somatic embryogenesis is, and you can see the full title at the top. We abbreviated SE, and this is like the only definition I'm going to give you in the whole talk. It's a process we use in tissue culture where we propagate a lot of structures that look like embryos, but like the embryos in seeds, except they are not from seeds. Essentially, what we do is we start with one embryo from a seed and get it to clone itself indefinitely. And this is a very powerful propagation technology. We can make thousands and thousands of copies of that original embryo. And in some cases, we can take a piece of a mature tree and pool that into making somatic embryos, but it's very rare. We've done that once in the lab that I know of with sweet gum, which who cares? Sweet gum. Well, and then you can take these structures, somatic embryos, which are all copies of the original seed embryo, and you can germinate them to make plants called somatic seedlings. And then you take them out of culture and harden them off and grow them just like they were seedlings, okay? So it's a clonal propagation technology, and it has a lot of applications. This is what an embryogenic culture usually looks like when we're working with it. It just proliferates and grows like crazy, and you'll see more of this stuff. We call them perembryogenic masses, and these are a stage that eventually makes these embryos. But at this stage, we can do all kinds of things with these cultures, and I'll show you those applications. Any questions so far? Okay, good. So what are some of these applications? One is, I've kind of given it away, we can use in vitro culture and specifically somatic embryogenesis to mass clonally propagate trees. We can do that for commercial applications, and there were companies that did this, but they're out of business. Like Arborgen, well, they're out of that business anyway. And another one in Canada. But most of these companies have found out it's expensive to do, and they'd rather just make seedlings from improved material. But you'll see in my lab, we have concentrated for the last 15, 20 years now on working for forest restoration purposes using somatic embryogenesis. And this is really good, a really good approach for propagating a lot of stuff up. It's also great target tissue for genetic engineering, and I have two examples of that to show you. In fact, most of the talk is going to be about that. And then also we can conserve germplasm very easily because once we have an embryogenic culture, we can take that and treat it and get it into cryo storage, and it'll stay there forever until we want to thaw it out, as long as we don't run out of liquid nitrogen. So these things are all very, very useful applications for embryogenic cultures. There we go. Now, I had to, I've been here 41 years, and I had to narrow down what I was going to tell you about today. But just to give you some idea of all the things we've done, I just made this one slide, okay? So we've used SE for clonal propagation of both hardwoods and conifers. Okay, it works for both. And then we've applied somatic embryogenesis to make transgenic trees for heavy metal phytoremediation, which is one of the big topics today. For conservation and restoration of threatened forest trees, which is another one, and for biomass energy. And the only example I have, if you see those really tall sweet gum trees there, those are hybrid sweet gum clones that we made. There's a planting of this out at Whitehall, and we've got some clones that just grow faster, much faster than the best American sweet gum. So that's what that's showing. And then some of the conifers are there, Atlantic white cedar, hemlock culture over on the far right in the middle. And then have a project that's over now with Kamal's lab, where we were propagating ash trees by somatic embryogenesis. That's an embryogenic culture on the left, and our planting out at Whitehall from these cultures on the right. And those are being screened for emerald ash borer resistance. So part one of the major things I chose to tell you about today is something that we haven't worked on for over 20 years now. And it involves engineering trees for what's called phytoremediation, okay, for cleaning up heavy metal contamination in soils and water. So a little bit of terminology. Heavy metals, we're talking about lead, cadmium, mercury, arsenic, chromium, and maybe a few others. Site remediation, for the most part, has always been an engineering approach. That is, dig it up, haul it away, bury it somewhere else. That's one kind of remediation. Or perhaps encase it in concrete, but didn't involve biology. But several decades ago, scientists started developing bioremediation, which mainly involved using bacteria that could break down contamination or process it and make it less dangerous. More recently, but not much more recently, scientists came up with applying plants for cleaning up heavy metal contamination. And that is called phytoremediation. I think I have it defined on the next slide. And so, and then finally, there are some plants, and including some trees, that will naturally take up heavy metals out of soil and water and process them, or at least accumulate them so they can be hauled off and something can be done with them. Okay. Now, there's the definition there. Phytoremediation is the use of plants to stabilize, reduce, or detoxify pollutants. Why would you use plants instead of bacteria? You just sprinkle bacteria on the ground, that would take care of it. Well, plants, including trees with their root systems, will suck up a lot of water with contamination in it. And they handle a lot of mineral compounds that are in the soil already. And so some of them can handle heavy metals. They will also, while they're doing this, they'll stabilize the soils and sediments against erosion. You don't have to do anything with them because they harvest all their energy from the sun, doesn't take any energy, and they help restore structure to the soil and organic matter. So this was, you'll see who gave me this list in just a minute. That's why we use plants. Mercury is a special case, okay? You may know that there are, or not, I didn't know this until I started working on it, there's thousands of square miles in the US alone that are contaminated with mercury. It comes from all kinds of sources, but my collaborator used to tell me there were huge electrodes that were used for different things that were filled with mercury, and when they'd crack, all the mercury would run out and into the ground. So there are places like that, especially in aquatic environments. The most toxic form of mercury compound is formed, which is methyl mercury. It takes Hg2 as ionic mercury. And then what happens in aquatic environments is that it's converted to methyl mercury by anaerobic bacteria. Methyl mercury is mobile and it's magnified in the food chain. That means that the first animals that ingest it, they get eaten by bigger animals, and the concentration of mercury grows at each level of the food chain until there was a disaster in Japan where people were eating fish from Minamata Bay. They had mercury and these people got mercury poisoning. It's horrible, and it causes neurological generation. It's a neurotoxin, okay, and it's untreatable. Now, one problem is that there are no plants that naturally accumulate mercury. There are no natural hyperaccumulators. While we're on this slide, does anyone know who that guy is on the right? The Mad Hatter. You know why people use the phrase as mad as a hatter? That's exactly right. Hatters who made felt hats would take pelts, and to separate the fur from the skin, they would soak it in mercury. Okay, they worked with mercury every day and they developed tremors and horrible neurological symptoms. So that's where the name came from Alice in Wonderland there. That's the Mad Hatter. Does anyone know who the little character is just off the Mad Hatter's elbow? He's probably poking him in the eye. You want to know who that is? No? All right. It's the dormouse, okay? So I have a special seminar trivia question for everybody that we'll answer at the end. Don't shout it out if you know what it is. It's all probably only old farts like me who's going to know what it is. The question is, and we'll bring this up at the end, what did the dormouse say? Keep that in mind. You don't have to listen to the rest of the seminar. Just think about that question. It turns out that bacteria that live in mercury contaminated soil have genes for mercury decontamination. They can take different forms of mercury and eventually convert it into elemental mercury, like mercury that used to be in thermometers. And you may or may not know this, but if you leave a puddle of mercury on the floor in your lab where you've spilled it and you haven't reported it yet, it'll evaporate eventually. It volatilizes at ambient temperatures, right? Okay. So what these bacteria can do, especially with MER-A and MER-B, and I'll show you the reactions, they can take methyl mercury, MER-B, you convert it to ionic mercury, and then MER-A converts it to mercury metal, which volatilizes. So it's much less toxic and much less dangerous than the other forms of mercury. Here's the reaction. You can see MER-B takes methyl mercury to ionic, and MER-A in turn takes the ionic mercury to Hg0, which is mercury metal. So here's my other mentor and collaborator that made a big difference for my career here. This is Rich Meager. Some of you probably know him. He's now retired, but he's still working in his lab as an emeritus professor over in Davidson Life Sciences. His idea was, let's engineer MER-A and MER-B into plants and have them phytoremediate mercury, since there are no natural mercury hyperaccumulators. The problem was that the codons, that is the 3 nucleotide sequences that code for amino acids in bacteria, are not the same ones that plants use for those amino acids. So if you just took the bacterial gene and stuck it right into plants, it wouldn't work very well. Rich is so smart, he figured out, oh, let's just modify those codons so they're plant-friendly codons instead of bacterial codons. And he used, and don't ask me to explain this because I didn't even understand it when we were doing this work. He used overlap extension PCR to change different percentages of the gene to the right kind of codons, okay? All right. You'll see one gene we use, it says MER-A 18. That means 18% optimized for plant expression. Okay. So Rich came up with this, and the first thing he put it into in his lab, and I don't know, Dayton, were you involved with the Arabidopsis stuff in his lab? Yeah, I thought so. And they put it into Arabidopsis, which was easy to transform. And then they got the little seeds, transgenic seeds, planted them out on plates of medium with ionic mercury in it. You can see the control is here. And on one side is the MER-A, and the other side is wild type. This is no mercury in this. But on 50 micromolar and 100 micromolar ionic mercury, you can see all the wild types died, but the MER-A ones are happy, very happy. So, you know, are you going to go throwing Arabidopsis out to phytoremediate sites? Probably not. And so I happened to be walking by his lab one day. I don't know how this happened. And he goes, Scott, come in here. I don't even know why I was in that building. It was the old biological sciences building where he was at the time. And I came in and he says, oh, I've got this idea. Do you have any trees we can do genetic transformation with? And I went, well, we do have these embryogenic yellow poplar cultures. We could transform them. Yeah. And he went, oh yeah, let's do that, okay? So there's those permogenic masses, and we could use those as target material for transformation. Now, I'm cutting out a whole lot of the story where Dayton and I failed to transform protoplasm. Remember that? But Rich at the time he'd been to some meeting and someone had introduced the gene gun. Anyone ever seen a gene gun? No, we have one in my lab. Okay, and what you can do is instead of using protoplasm or using agrobacterium, which I'll show you later, you can actually coat tiny little metal microparticles, gold or tungsten, I think was the other kind, with DNA, and then blast them right through the cell walls with a gun into the cells, and some of that DNA will get off the particles, get into the nucleus, and incorporate into the chromosomes. So you can get genetically transformed material. this is the diagram of, I'll walk over to that side later. But how it's done, we have an embryogenic suspension culture. We would dump it through some screens to get little cell clusters, and then we bombard it with the gene gun. And the early version of the gene gun actually used a 22 blank. It was dirty. We had gunpowder all over the place, and it showers these particles down onto the embryogenic cultures, and then we grow them up under selection to get the transgenic material. This is the current version of the gun. It uses compressed helium, which is much cleaner, but that's why it's called the PDS 1000 AG. It's a Biorad product. I think Biorad still owns it. I don't really know. Okay, so that's when Dayton came into the lab, and he was the first person in the world to transform a forest tree using microprojectile bombardment. And we published this work. We had a reporter gene on a plasmid we used called GUS that you can easily check for expression of with a little bit of a chemical you add to it. It turns everything blue. And there's a 4-year-old tree with the leaf coming out of the bud, and if you stain for that, it turns blue. So we knew we could transform yellow poplar this way. And look at that kid. He was so young. That was Dayton when he was working in my lab with me and Rich. Well, when Dayton moved on, Clayton Rue was a PhD student in the lab, and we decided, okay, we know we can do this. Let's try the mercury genes. So Clayton used the microprojectile bombardment to bombard yellow poplar cultures, and then we made little somatic embryos and germinated them on ionic mercury. And you can see the transgenics are germinating, their little roots are extending, and the controls dry up like little match heads. So we could get these transgenic yellow poplar Mur-A trees that could take ionic mercury and convert it to metallic mercury. This burned us when people still read paper journals. This got us on the cover of Nature Biotechnology. And we were all very, very proud of that. But most of the real methyl mercury problems are in wet soils, right? Well, yellow poplar doesn't like wet feet. Eastern cottonwood does. So Rich is going, well, you got anything else? And so we got some cottonwood clones, I think from Arborgen, cottonwood cultures from Arborgen, and transformed those. They were not embryogenic. What they were, though, they could do is make adventitious shoots from leaf pieces. And so Dongsheng, who was a PhD student in the lab, transformed Eastern cottonwood trees with Mur-A and eventually with Mur-B as well. And here's his results here. You can see here are 2 of our Mur-A clones, and then leaf pieces from wild type, which is bleached white on this level of mercuric ion. Now we can grow up some trees and do something with them. But remember I told you that mercury is a vapor at ambient temperatures? How could we tell that it was working? We would hook up our trees, this is a yellow poplar, to a mercury vapor analyzer and measure the amount of mercury that these trees were processing, and then it was volatilizing out of their stomates, out of the leaf stomates. And when we did that, this is what we saw. The bottom line is wild type, and over time you can see the Mur-A18 is making a lot of mercury vapor. So that was working. It's pretty good, huh? Gee, people are going to go wild over this, aren't they? This looks even more impressive. Look at it. These are, we purposely contaminated potting mix with mercury ion, and the wild types on the left, the rays are on the right. These actually did get planted out. Guess where they planted them? On an old hat making factory site in Danbury, Connecticut. And the community, the city council approved this. I guess they got APHIS approval. I wasn't involved with it, but they planted these trees out, and then nothing. Essentially, Rich started 2 different phytoremediation companies, thinking that these would make him rich and famous. The problem was, 1, people didn't want transgenic trees very much at that time. They didn't like them. 2, companies who were responsible for polluting found it much easier to just hire lawyers to keep everything tied up in court than to pay us or pay Rich to make transgenic trees. I only ever got 1 bit of data out of this that I saw. It wasn't my work anymore. And it showed that the trees in the ground actually were processing the mercury, because the wild type accumulated the mercury in their tissues, whereas these trees, the transgenics, blow it out of their leaves, so they have lower internal mercury. That's as far as that got. I'm sorry to end on a kind of a down note, but that's where it stands. Even Rich doesn't know what happened to these trees because I'd asked him more than once, and he's not sure if they're there anymore or what. Okay, any reason to say more about that story? Let's go on to the other story. How am I doing on time? Okay, good. So part 2 is going to be American chestnut, of course. We have worked on a number of trees that are in big trouble, threatened trees. We worked on American chestnut. We worked on Carolina and eastern hemlocks. I think I showed you the photo early on. Ash species. We've worked on red bay, swamp bay, and sassafras that get laurel wilt, and Atlantic white cedar, which just got all cut down and its habitat destroyed, but it makes really pretty little plants. So why would we want to do this for these threatened trees? One is clonal propagation of good genotypes that are resistant to these diseases and pests already, to help conserve genetic diversity because we can do the cryo storage, and as target material for gene transfer. Okay, so perhaps we want to put genes in for pest or pathogen resistance. We can do it that way. Usually it's for testing candidate genes, but the chestnut has gone further than that into possible deployment sometime in your lifetimes, maybe not mine, but okay. So here's American chestnut growing in the Smokies around 1900. It was the biggest, fastest growing tree on the ridges and benches in the Appalachian Mountains, making its best development in the mountains of North Carolina, Southwest Virginia, and did pretty good in the Georgia mountains as well. Those are all American chestnuts. The yellow poplars are the skinny ones in the background. That's the only tree that could compete with it up in the mountains. 100 feet tall, 6 feet in diameter. If you cut it down, the sprouts would grow even faster than the original tree, 6 feet a year. Very aggressive and would dominate sites, just take over sites because it would just keep sprouting. Even now, you go up in the mountains where they've done a clear cut recently, the first things out of the ground are chestnut sprouts that come up. Chestnut was very, very important to all the people who lived in the southern Appalachians. Every structure you see in this photo, including the fence, is all chestnut. That cabin there is chestnut timbers, chestnut siding, chestnut shingles. The little building there, they're heating their water to wash the clothes with chestnut. It was used extensively. The reason you see it like that is because chestnut is naturally durable wood. That means rot resistant. It has a very high tannin content, and you can use it for anything on outside or that's in contact with the ground, and it will last forever. No, not forever, a long time, but that's what they use pressure treated lumber for now. Chestnut was naturally rot resistant. Very important. I could tell you tons more about this, but we'll certainly run out of time. But I do want to tell you about the nuts because we have another part of our story here. The nuts themselves were very different from the nuts you think of in the forest, like walnuts and hickory nuts and acorns, in that their storage product is starch. It's not fat, which is what most nuts have. It's starch. And that's why if you've ever eaten a chestnut, it tastes so different from other nuts. But very nutritious, high quality food for wildlife. And it's said, although I've never seen documentation of it, but the old timers said that all the wildlife was more abundant and grew to larger size when the chestnuts were here. They're in spiny burrs with 3 nuts per burr inside. And the great thing about them for wildlife and for people was there was never a nut crop failure because chestnuts don't flower until June. All those other trees flower earlier, and if there's a late frost, boom, their whole nut crop is gone. Chestnuts were always there, and every year there was a big crop of them. Wildlife would feed on them. The people who lived in the southern Appalachians would drive their hogs up into the mountains to feed on the nuts on the ground in the fall, to fatten them up before they'd sell them. Very, very important to people throughout the southern mountains. And even in cities, this is actually in Philadelphia, it's called Fairmount Park. People would go out in the fall and go chestnutting. They come out with picnic baskets. People would climb the chestnut trees and jump up and down on the branches, and the nuts would come raining down. People would pick them up, take them home, roast them, and eat them. So very, very important. Now, here's Mel Tormé. Does anyone know Mel Tormé's nickname? Ever heard of Mel Tormé? Most people haven't, but you have heard his song, Chestnuts Roasting on an Open Fire, the Christmas song. He wrote that. He wrote that song. his nickname was the Velvet Fog because he was a singer-songwriter. So that's how Mel got into the sound. Okay. Now, do you find it interesting that two of the most popular Christmas songs ever were both written by Jews? Mel was Jewish. Who was the other? Anyone know? Irving Berlin wrote White Christmas. It's true. Well, all this came to a crashing halt in 1904 when a forester working at what's now the Bronx Zoo in New York City noticed that the chestnut trees lining the streets there were dying. And what he found on them was this yellow pustular stuff. He took some samples off and went to an acquaintance of his at the New York Botanical Garden named Merkle. Oh, by the way, this guy's name was Merkle. The forester's name was Merkle, but it was Merkel, not L-E. So no relation to me. But anyway, he took the samples over and Merkle identified it as a pathogenic fungus. He called it Endothia parasitica. About 30 years ago, it was renamed Cryphonectria parasitica. And what it does is it gets on these trees, forms through any crack. They will grow through any crack in the wood, like a broken branch, or even when the bark cracks, and get under the bark and start killing the tissue underneath. It's a necrotrophic fungus. It puts out oxalic acid. So it secretes oxalic acid that kills the tissue and then it eats the dead tissue. Sounds like a good idea for a TV show. In any case, it makes this sunken canker that eventually girdles the tree dies. How did it get here? It came in on Chinese and Japanese chestnuts that people imported because they were smaller stature trees. They could grow in their yards and they could pull off chestnuts instead of hiring people to climb the trees and jump up and down on the branches. So this fungus was an Asian fungus. It came over with the Asian trees, got off onto the American chestnut trees, which had never been exposed to it before, and just started wiping them out because they had no natural resistance. These are some of the scenes from what happened. That's an entire mountainside of dead chestnuts. It made what are called ghost forests. These trees, they would die, the bark would fall off, but the trees would keep standing there because they were rot resistant, and then they'd eventually fall over. While they were on the ground, they got infested with larvae of, I don't know, some moth, I think, and people called them worms, and they made holes in the wood, which was later salvaged and cut up into timber for furniture. It was called wormy chestnut. It's very desirable. How many of you all have ever noticed that when you walk into Warnell, there's all this paneling with lots of little holes in it? I don't know how many years I walked through there and just went, oh, I wonder what those holes are. It's wormy chestnut. This building was built in 1939. I think it was finished in 1939. Right about the time chestnut blight got to Georgia, killed a bunch of trees, and there was walnut timber, chestnut timber to use. Dr. Joe Dolan took a sample of this from above the drop ceiling and analyzed it and confirmed it is American chestnut.
That project is still.
Yes,
in
I know.
progress.
I know what you mean. We'll talk about that later. So what did they try to do to stop the blight? I can't talk about hardly any of these, but in New York City, they tried spraying the trees with fungicide, and that didn't work at all because it was under the bark. They tried quarantining it by trying to stop it before it spread further south. They tried to find resistant American chestnut trees, and there are some that have some resistance, it turns out. They tried gamma irradiating nuts, hoping to get mutations for blight resistance. Hypervirulence I can't cover, except it's a virus that actually infects the fungus and makes it weak so the trees can actually grow through it. The other things I am going to talk about a little bit, the Chinese chestnut is resistant, and it can be bred with the American chestnut to make resistant hybrids. And finally, what we do in my lab is the in vitro propagation and genetic engineering. These are some of what we call large surviving American chestnuts that get the blight, they get cankers, but then they heal over. Doesn't kill the trees, and they get pretty big, and they make nuts and everything. They have some low levels of resistance, and we do work with these trees in our lab. We propagate them. And I've actually supplied a lot of trees to people to test. This is the American Chestnut Foundation's approach. They hybridize with the Chinese chestnut. This one's out at the corner of College Station and Barnett Shoals Road. There's 3 chestnut trees growing there in the big new parking lot they built. I got them to save those trees. They were going to cut them down and just build a parking lot over them. But the grounds guy, Brett Gannis, he said, okay, we'll save them. He did. But what they did is they hybridized, and you can see the F1 hybrid is half American, half Chinese, and then they back crossed again and again to American to get trees that would look like American trees, but then they would inoculate them at each step to see which ones were resistant to blight, and they picked those for the next generation. Finally, ending in this BC3, backcross 3, F3 generation that was supposed to be mostly American with the blight resistance of Chinese. The only problem was they thought there was only 2 or 3 genes involved in blight resistance when they started this, and it turns out there's like 20. So they missed a lot of the genes, and these trees are not as resistant, any more resistant than the F1 hybrids. So it's kind of sad. But they have gone back now, they are retooling their breeding program. They've got molecular markers for all of these blight resistance loci, and they're going to come up with trees that look pretty darn good and have pretty good blight resistance. But I can't go into that. But what about us? What I started doing in 1989 was, oh, I can just culture chestnuts like I do yellow poplar. That was like the stupidest thing I've ever done. Because chestnut is the biggest pain in the ass to work with of any tree we've done in the lab. The photo right up here, this is a newly initiated chestnut culture. We take the little seeds out of the nut and put them on media, and I'll show you the media in a minute. And all those blobs coming out are little somatic embryos. And then we can move that onto media, and it'll keep making the somatic embryos. It took us until like 9 years to get the first ones to actually germinate and make plants. And then finally, we got some out in the field years after that. It was a long process figuring out how to get these to work. And it's only in the last few years we've gotten better at it with Heather and Ryan in the lab working on improving the culture system. Just in case you're curious about what's in tissue culture media, it's fertilizer, top line there, major elements, minor elements, iron, vitamins, sucrose, because these things aren't photosynthesizing. They have to have some carbohydrate to live on. One amino acid above all the others works, glutamine, and then 2,4-D. Who knows what 2,4-D is? They're all foresters. You must know what 2,4-D is. But you're not all foresters, are you? 2,4-D is a herbicide. It kills plants. That's what it's like Roundup, except Roundup's a different chemical. Used at part per million quantities, that's 2 or 4 parts per million. That's what triggers that original embryo that we start with to start cloning itself. And it works with all kinds of plants, soybean, corn, conifers, and with trees. That's what's in media. So we worked for years scaling up this thing because it was difficult to work with. And it turns out they grow much faster in liquid medium instead of gel medium. And we can make lots of embryos, and we put them in the refrigerator, give them a cold treatment before they germinate. And we're lucky to actually germinate in the cold. And then we can put them in magenta boxes. That's activated charcoal in the medium to protect the roots from getting light on them. And we can make quite a lot of trees, but those last 2 steps are the hardest ones. We can make tons of embryos, but getting plants all the way through to the greenhouse was really, really tough. We're better at it now than we've ever been, but we're still not great. But that's what we did. That was 10 years of work there. The cryo storage. This is Paul Montello. I think Paul is here today. He is our cryo, I put wizard there. You're really the cryo guru, aren't you? Whatever it is. But anyway, we can take little samples of the embryogenic cultures, stick them in liquid nitrogen, and we can pull them out. I think our record is 15 years later, but you can leave them in there hundreds of years if you don't run out of liquid nitrogen. And when they come out, they look like hell. But you can see that after a few weeks, they start recovering and regrowing, and eventually have your culture back. This doesn't work for every single culture. I think with chestnut, we've been as high as like 80% recovery at times. But it's nice that we do it because we have thousands of these cultures. There's no way we could maintain them if we didn't have cryo. Okay, what about the gene transfer? Up in the upper right-hand corner is the person who's really responsible for developing the system. Gisele Andrade Dean is still kind of at the University of Georgia in Griffin. Many of you have probably met her husband, Jeff Dean, who is the Ag Station director there. Gisele is married to him, and while they were here at UGA, this part of UGA, she worked in the lab and developed this transformation system. It uses agrobacterium for the transformation, a natural genetic engineer soil bacterium that will move DNA into plant cells. And this is the selection process. We use antibiotic selection, and you can see these little yellow blobs here are ones that have the antibiotic resistance gene. So these are ones that are transgenic. And then when we stain for Gus, they make blue clusters. We can pick individual ones of these. The blue ones are dead, but we can pick other ones that are growing, grow them up, make embryos that turn blue. And if we took those plants, they would turn blue. That's another 10 years of work there. This was really, really tough. The liquid selection was the only thing that would work. If we just tried growing them on plates of selection medium, we got all kinds of stuff growing up that wasn't transgenic. The liquid is very stringent, because they're bathed in the antibiotic, and we can get the transgenics out of there. So what genes could we put in once we had it working? Most of them came from Chinese chestnut. People had various ideas about, oh, I think this gene's involved in the resistance. And there's ways I could tell you that they determined this. But a lot of the genes came from other plants that had been shown in other systems to confer resistance to fungal pathogens. One from an orchid, one from grape, one from pepper. The only one that's worked to some extent so far is the wheat oxalate oxidase gene. And this was not at all my idea. This was someone else's idea. Dr. Bill Powell, who's passed about a year ago, he had the idea. He read about oxalate oxidase. He said, oh, gee, what is it that kills the tissue under the bark that chestnut blight uses to kill the tissue? Oxalic acid, remember that? What if we could use that and break down the oxalic acid? That's what oxalate oxidase does. Chestnut doesn't have its own oxalate oxidase, but wheat does. Okay, let's put the oxalate oxidase gene in and into the tree and see what happens when we inoculate it with these strains of the fungus. And that's what EP155 is. That's a chestnut blight strain, a virulent one. And you can see there's pure American chestnut, and you can always see the sunken canker forming on that guy. That's Chinese chestnut. It's made a little canker, but it's starting to heal up, so it's got some resistance. Here's what he calls the Darling 58 tree. It's the transgenic oxo, and you can barely tell that the wound is there. So it would seem to be working. Why is it called the Darling 58 tree? Herb Darling was the president of the New York chapter of the American Chestnut Foundation that gave Bill's lab hundreds of thousands of dollars to support their research. They had a lot of millionaires in their chapter, and his lab was supported very well, so he named that transgenic tree after Herb. Well, what's happened since then? I can't tell you all that's happened since then, but I will tell you that mistakes were made. And right now, the American Chestnut Foundation, which was also supporting Bill's lab, was not happy with how they were being treated and some other things that happened with the Darling trees. And they have come to us in the last 2 years and said, you know, we want to find out really whether oxo is really good or not. And they asked us to start new American chestnut cultures because we had stopped doing transgenics a couple of years ago and transform them with our own oxo genes. And that's what we've been doing. You see Ryan and Heather there. We started new cultures, not just from one New York tree, which is what the SUNY group did, the SUNY ESF group. They were going to put, or they did, they put this gene in one New York tree, and then they were going to let it grow up, make pollen, and pollinate all the other American chestnut trees with it, so that they'd all be related back to a single New York tree. Not a good idea. So we started trees. We started cultures from Pennsylvania trees, Georgia trees even. You see that one at the bottom. And tested them, make sure they make trees, and then started transforming them. This is the vector we're using, the PFHI OXO1. Here's the OXO gene, the yellow guy, being driven by an Arabidopsis ubiquitum 11 promoter instead of a cauliflower mosaic virus 35S promoter, which is what SUNY used. And with a lot of hard work from Ryan and Heather, we've got our first transgenic OXO trees. You can see that there's a nice rooted one there, and there's a bunch of them in pots. PCR evidence is there that the gene is in, and we can even do something, and this isn't Gus, we can actually feed these plants a little bit of oxalic acid. We wound them with a needle. See the leaf there on the left and the leaf on the right. The one on the left is just one we've wounded with a needle without any oxalic acid. But then when we do another one and put oxalic acid on it, it makes this very dark blue-black stain. It's almost like having another reporter gene. So this is making the enzyme. That's evidence of the enzyme oxalate oxidase activity in these trees. Okay, I'm still on time, am I? Or have I run over? Still good.
15 minutes.
Excellent. Okay, because we have a special trivia quiz to do still, right? Okay. So these, you've seen most of these people already, and I've talked about them. Ryan has been working with me for 15 years. Heather for not quite that long. They are the people who are doing almost all of the chestnut work, although we do have 2 undergraduate students doing chestnut work right now. Caroline and Julianne were undergraduate researchers in the lab, working on the chestnut, OXO chestnut actually. Caroline was also working on Ozark chinkapin, which we've also got embryogenic cultures from. Ryan McNeil is doing some work for his PhD on chestnut in my lab. Emma is actually working on ash embryogenesis and will finish her master's thesis on that subject early this summer. I can't show pictures of everybody who's been through the lab in the last 4 years, but these are people who are here now, and some of them have been here a long, long time. Paul, how long have you been working with me?
Since the 90s.
So that's why, look at his hair. He was a young guy, and now look at me, look at me. Okay, so it's been a long time, but Paul does all kinds of stuff in the lab besides the cryo. Okay, and then I also can't name all of the supporters of my research over the years, but the current supporters, the American Chestnut Foundation, I've been very, very happy with their support. We work very closely with them now. Foundation of the Carolinas actually funnels money through them for us to use. This fellow named Brad Standback, who that's his foundation. He's been great. The Schmidt Foundation, Horticultural Research Institute, Stanley Smith Horticultural Trust, mainly have supported the ash work, and the Forest Service has been supporting work with Oregon ash that we're doing now. Okay, then, who's got the answer? What did the dormouse say? Bob Cooper, of course.
Feed your head.
Exactly. You've hit the nail on the head there. That is it. Thank you. Thank you. No one's ever answered that correctly. And I didn't give this talk that many times on that subject, but you got it, Bob. So if you were like of our generation, you would know this song, White Rabbit. It was the seminal, I think, psychedelic rock, acid rock is what it really was, song of the 60s. And Grace Slick was great. Now she's a crazy old lady, but she was a great singer. And that's actually, that was a trick question, because did the dormouse actually say anything in Alice in Wonderland? You know what he said, Cindy?
Something about might as well, something about you say that it's the same to say that you sleep when you breathe and you breathe when you sleep or something.
It was like nonsense because he was asleep most of the time, right? He was a dormouse. He was dormant, right? So he actually did say something, but most of the time he just sits there and he's sleeping. Okay, well, that's my last slide, and I'd be glad to answer questions or whatever else you all want to do.